was relevant local and global problems. The Institute has several units. I'll just mention some of them. We have the Epidemiology and Biostatistics Research Unit. We have the Neuroscience and Aging Research Unit, the Drug and Infectious Research Unit. We have Genetics and Cancer Research Unit, Phytomedicine Unit, Palliative Care, to just mention a few of them. Over the years, the research of the Institute has increased tremendously, and the facilities of the Institute are utilized by postgraduate and occasionally undergraduate students of University of Ibadan, other universities and polytechnics in Nigeria. The Institute is headed by a director, and the current director is Professor Ekeluapo O. Ajayi. And I would like to hand over to her now so that she can give her opening remarks. Thank you very much. Professor Ajayi.
Thank you very much, Dr. Adeniji. I welcome everyone to this February webinar of the Institute for Advanced Medical Research and Training. Well, it's been a good year for us, and we are very happy to also have Professor Fowotade as our speaker today. Professor Fowotade is a global figure, and we are very proud of her in the College of Medicine and the university too. And today, I believe what she has to share with us is something that is global and would also give us some insight into virus diseases and how the scientists have been able to. Just a minute, please. I did. Okay. Can you hear me now, please? Can you hear me?
Yes, you can hear you, ma.
Oh, okay. Thank you very much. I welcome Professor Fowotade, and I thank her for accepting to give this lecture, February lecture, as one of the series for the Institute for Advanced Medical Research and Training. Professor Fowotade is a global figure when it comes to virology, clinical virology. And she is a pride to the College of Medicine, the University of Ibadan, and I think Nigeria as a whole. Professor, you will hear more about her profile, but for her to even have time for us is really well appreciated because she's very busy. Today, she's going to share with us issues about virus diseases, how scientists have been able to get around it using genomics. And genomics is actually getting to be the in thing now in diagnosis.
Good afternoon, everybody. Can I ascertain that you can hear me clearly?
We can hear you, but I'm still speaking, ma. Prof, ma. I hope we can hear you, Professor Fowotade. But can you hear me too? Others, please? I don't know. Can you hear me?
Yes, ma. We can hear you.
Hello, am I audible?
You are also audible, Professor Fowotade, but we are introducing you, so please hold on. We will start the lecture soon. So tonight, this afternoon, she's going to tell us a bit about virus hunting. which is, they are all in the highlights of the lecture: virus hunting, genomics in action, lab to field impact. This is very important because we talk now about translational research, translational science, knowledge transfer, and so on. And this means that whatever we do in the laboratory, we should be able to take it to town and to impact on diseases, preventing and curing diseases, and improving public health. So this is a very topical issue, and we know that a lot of viruses has actually affected countries and even globally. We have the Ebola virus, we have the COVID-19, and she has actually been part of all of this. So we hope that at this lecture, she will also share her experience about this. So I welcome Professor Fowotade, but a citation will be read just before she gives a lecture. Thank you very much for joining us this afternoon. Thank you.
Thank you, Director, for that short introduction. We would like to invite Dr. Bakari to give the citation of our lecturer of today. Dr. Bakari, you have the platform.
Thank you very much, sir. Can you hear me?
Hello? Yes.
We can hear you.
Yes, we can.
My name is Dr. A.S. Bakri. I'm to read the citation of Professor Adeola Fowotade, our guest lecturer for today. Adeola Fowotade is a professor of clinical virology in the Department of Medical Microbiology and Parasitology at the College of Medicine, University of Ibadan, and a clinical virologist at University College Hospital, Ibadan. She currently serves as the Provost of the Benjamin Carson, that is Senior College of Medicine, College of Health and Medical Sciences, and holds a Bachelor of Medicine and Surgery degree from University of Ilorin, Nigeria. Additionally, she holds a doctorate degree in molecular virology and immunology from the School of Biosciences and Medicine, University of Surrey, United Kingdom. Professor Fowotade is honored as a fellow of the Faculty of Pathology from both the National Postgraduate Medical College of Nigeria and the West African College of Physicians. Her exceptional talent is further recognized by holding the esteemed GVM Rising Star Mentorship Fellowship. With a unique background and training in virology, immunology, and epidemiology, Professor Fowotade developed a transdisciplinary and translational research program at the University of Ibadan that focuses on viral immunopathogenesis and the molecular epidemiology of viruses and their impact on global health, human health. Our research program focuses on oncogenic viruses, hepatitis B, and human papilloma viruses, as well as emerging and re-emerging viral infections, including Lassa fever, coronaviruses, and currently monkeypox. In addition to a track record of productive research work and publications, she is frequently called upon as advisor and conference speaker at national, regional, and international level. Professor Fowotade was a member of the Oyo State COVID-19 Task Force and also the laboratory pillar lead for the Oyo State COVID-19 Emergency Operations Center from the year 2020 to 2022. She coordinates a molecular biology laboratory accredited by the NCDC for SARS-CoV-2 and Lassa fever genomic surveillance. Professor Adeola Fowotade served as a consultant to the National AIDS FWACP and on the National Clinical Mentorship Program on HIV/AIDS, and is currently a technical advisor to the WHO on. Professor Fowotade has 85 manuscripts published in esteemed international journals, including Lancet, nature, and science. This is fantastic. Please welcome our guest lecturer for today, Professor Fowotade, as she presents our lecture this afternoon. Over to you, Madam Provost, Professor Fowotade. The floor is yours now. Thank you. Hello, Prof.
Professor Fowotade, we are ready for you.
All right, good afternoon, everybody. And I want to thank Professor Adeola Ajayi, the director of IAMRAT, for inviting me to give this lecture. I also want to thank Dr. Bakri for the kind introduction. This afternoon, I'll be speaking to you on a subject titled From Genome to Global Health, Translating Viral Genomics into Public Health Action. And the content of these slides details only an element of my research trajectory at the College of Medicine, University of Ibadan, at least within the past 5 years. I'll follow this outline. I will start with an introduction to the evolution of virus hunting, and then I'll speak to translational genomics in action, using the mpox virus as a specific example. And then I'll conclude by talking about preparedness for the next pandemic.
Professor Fowotade, please check your
If I take you back in history to how viruses evolved and how they were detected, in the pre-1800s, viruses were mainly regarded as invisible agents, more often as even mysterious agents.
Your slide is not moving. Please check your slides and make it a slideshow.
Okay.
Yeah. Moving now, but you can make it slideshow. Use the slideshow mode, please.
Okay, just to be sure, can you see my slides now?
We can see, but not in the slideshow.
Maybe
mode.
I need to take off my camera to ensure that at least the slides are working well. Sorry, can you let me know if you can see my slides now from the screen? What's going on?
Yeah. Professor Fowotade, please. Can you just start all over, and your slides are fine and now in the slideshow mode.
All right, thank you very much. I've now been able to unmute. It looked like some alien viruses are taking over my slides, but I'm in control now. My sincere apologies. All right, so I will be following this outline. I'll give a brief background to the evolution of virus hunting, and then I'll talk about translational genomics in action using the Mpox virus as a specific example. And then I'll close by talking about preparedness for the next pandemic. As far back as the pre-1800s, viruses were just mere invisible agents. And to many people, they were mysterious agents, sometimes some kind of afflictions from the gods. Apologies to non-Yoruba speaking persons on this call, but you will recall the days of Shopana, the deity that was associated with smallpox, and Igbono as measles was known. And much later, Igbono Olobo as monkeypox was called. And then we moved on to the era of detection of discovery of filtrable viruses. Because of the small sizes of viruses, they were mainly known as filtrable agents because they could be trapped in small micron filters. And that was all. They could not be visualized. But when we moved to the cell culture and electron microscopy era in 1940s to 1970s, viruses could be detected based on their destructive effect on living cell culture. And these were referred to as cytopathic changes. And sometimes when they were injected into lower animals, like mice, the lesions that they caused could also be observed. We entered into the molecular virology era between the 1980s to 2000, and that was like a eureka era for virology, because then we could use commercially available primers and probes in polymerase chain reaction, PCR, to be able to detect targets of nucleic acid of different viruses. By 2000 to 2010, we had entered the metagenomics era, and we had next generation sequencing, which allowed us to be able to detect novel, unknown, previously unknown viruses. And this was a good day for science because it was like a shot in the dark, giving you a lot of data that you could analyze and use to be able to detect even genomes that were not known to exist from the past. After the COVID-19 pandemic in 2020, up until the present time, we seem to have entered into the era of real-time pandemic genomics. And at this point, next generation sequencing is now being adapted to real-time genomic surveillance. We have portable Oxford nanopominum sequencers being used inside labs of clinics, of airports for real-time sequencing and variant tracking, even among travelers, particularly during the days after the lockdown was eased off. It was important to know what variants were being imported from what countries into other places. And this was used a lot in the developed countries. What has happened in the major paradigm shifts in virus hunting is that we seem to have moved from a period where all we had was symptom-based and we could not be able to even track the causative agents to an area of filtrable viruses where we had some proof of viruses that we could filter which we could not visualize. And then during the cell culture era, we could visualize the virus, we could isolate them, but that process happened to be slow and culture dependent, and so it was tagged unreliable. With the molecular biology era, we now had a more sensitive and specific platform for detecting viruses, but we still needed to have known targets, known primers that could be able to target the viruses of concern. With metagenomics, we could discover new novel viruses or previously unknown viruses. But then in addition, you also generate a lot of data that was complex and difficult to interpret. With real-time genomics being utilized for real-time genomic surveillance, we seem to have moved from an era of reactive surveillance to more predictive surveillance. Now with our epidemiological data, with our laboratory data, some mathematical modeling, we could do some predictive surveillance and even be able to tell when are we likely to have the next outbreak or the next pandemic, when will this occur. But even this requires some global coordination to make effective use of that knowledge. And so the question that comes to mind is, who is a virus hunter? That person that is able to detect viruses, translate genomics into data, and utilize it for public health action that feeds into global health. And so when I asked ChatGPT, who is a modern virus hunter? He told me that is a genomic epidemiologist who has some knowledge of bioinformatics, who can function as a field outbreak investigator, and ultimately plays the role of a global health defender. And it also gave me an image that looked like the kind of things that I do. If we look at the evolution of portable laboratories for detection of different types of viruses, we are moving from an era where we had to rely on laboratories with heavy equipment like the real-time thermocycler, the CFX96 by Bowrad, which can only be positioned in the lab, to an era where we have near point-of-care tests, which are also molecular-based, like the Cepheid Gene Expert, the QIAGEN CareStats, the SD biosensor, the DASH platform, and a lot more that were able to bypass the stage of nucleic acid extraction and have like an accelerated turnaround time that usually would span about 1 to 2 hours. And that sounded really good. But then came the portable Oxford Nanopore Minion sequencer that is about the size of a 10-inch iPad, which you could take with you into the field or even into site labs, and which is not only able to detect the viruses, the genotypes, but can also tell you the points at which the viruses have started to mutate. And this is a good day for science and a good day for virology. During the time of the COVID-19 pandemic, in collaboration with Professor Adeola of IAMRAT, we had, in a multi-country phase 2 study, evaluated a suitcase lab for rapid detection of SARS-CoV-2. And we also had collaborators from 6 other sub-Saharan countries. On the image that you have before you, you will see the suitcase lab. And in that suitcase is an isothermal instrument, which utilizes the principle of the loop-mediated amplification that allows you to be able to get your targeted nucleic acid in it. And everything that you need to make the system to be functional was also right there in the box. The centrifuge, the power source, the pipettes, even the disinfectants are all there in the box. We actually got different results from different countries. However, the most significant thing was that we seem to have been able to contribute to science in terms of being able to produce and evaluate the efficiency of a portable laboratory that could be used for real-time tracking of outbreaks. And so, why do genomics matter in outbreak investigation? This is the only way you'll be able to detect the outbreak much earlier than if you had to rely on traditional epidemiology or culture-based methods. It also helps to be able to identify hidden chains of transmission that would not have been discovered using the traditional methods that were in existence. Additionally, it helps to be able to differentiate between local transmission and imported cases, as we sometimes do have. And the data that you get from all this guides targeted public health intervention. What is the best therapeutics to be used? What is the best vaccine strategy to be adopted? In the days of this COVID-19 pandemic, the only way we knew that you would not be able to detect the Omicron variant with the existing diagnostics was because genomics had shown that an aspect of the sequence of that virus you would not be able to detect it with the existing diagnostics. And that tells you how much importance genomics contributes to outbreak investigation and ultimately to public health. In 2022, in collaboration with Professor Christian Happy of Redeemer University, we have been able to report the first 100,000 genomes of SARS-CoV-2 that were sequenced on the African continent. And our lab in Ibadan had contributed a total of 1,300 genomes to the 100,000 that were reported at that time. And this was published in Science as the evolving SARS-CoV-2 epidemic in Africa, insights from rapidly expanding genomic surveillance. And the data from that study was very useful as it served as a guide to the African Center for Disease Control on the appropriate public health intervention. It was also interesting to see the variants as they evolved across different parts of Africa, and also to compare what we found with what was also being reported outside of the African continent. Back home in Ibadan, in collaboration with Professor Moses Adewumi, we had looked at the multiple expansions of globally uncommon SARS-CoV-2 lineages in Nigeria. And we had found here in Oyo State that we had predominantly the Eta variant as well as the Alpha variant, rather than the Delta variant, which was predominant at that time. Interestingly, we also found that the Eta variant showed a much increased infectivity and decreased neutralization when you expose it to convalescent sera in vitro. And we're also able to discover, using phylogenetic reconstructions, that the Eta variant originated first from West Africa before spreading globally as a variant of concern in early 2021. Much recently, we had also published in the Clinical Infectious Disease Journal a collaboration with Professor Hiru Kaukeke of the University of Ibadan, a study on the genomic surveillance of SARS-CoV-2 in Ibadan, Oyo State. And in this study, our focus was on the healthcare workers. We were interested in the reasons why they were having breakthrough infections, despite the fact that they had been vaccinated. And it was quite interesting that in this study, we were able to identify a novel lineage that had never been reported in Nigeria. That is the B1575, which earlier than that time had been reported only in Spain, but not in any African country. And that study led to the fact that there was a need for more rigorous surveillance efforts to unveil potential low-level lineages that were circulating across different African countries. It was interesting that this new lineage that was found in that patient, who happened to be a healthcare worker, was a variant that happened to be associated with a lot of, much severity compared with other emerging variants at that time. And so it was also noteworthy that this healthcare worker had a complete vaccination with the Oxford AstraZeneca vaccine before having this breakthrough infection that was caused by the B1575 lineage. And so this leads to translational genomics in action. And I will be talking about emerging and re-emerging viral infectious diseases as they are found across the globe. The image that you have before you shows you that emerging infectious diseases, whether they are newly emerging or they are re-emerging or resurging or they are deliberately emerging, do not have boundaries. They are not localized to Africa. They are found in every part of the world. And in West Africa here, this part of the world is home to emerging and re-emerging Lassa fever, yellow fever, and a lot more other viral infectious diseases. In Central Africa, Democratic Republic of Congo, they have been persistently plagued with recurrence of Marburg, Ebola, and now even with Mpox. But why does this matter? We would find out in the next slide. Some of the lessons that we ought to have learned from COVID-19 and which should shape the way we would view contemporary outbreak is the fact that the world now is a global village. With travel, globalization, trade, and traveling, you can almost get to any part of the world within 24 hours. Regardless, infectious diseases also would be able to cross boundaries more easily than ever before. With mutation, microbial adaptation, and resistance, we now have more frequent outbreaks of emerging infectious diseases. And as human population continues to explode, and we now have humans moving into previously uninhabited area, we have closer interaction between humans and animals. And so we now start to have zoonotic infections as spillover infections in humans. Climate change by itself has not helped. And the human and economic toll that emerging and re-emerging infectious diseases have can be quite enormous. Therefore, responding effectively to these outbreaks has to be a global health approach that cannot be limited to just one country. At this point, I would like to talk about the 5 stages through which pathogens of animals evolve to cause diseases in humans. And you will understand why this is important as we move on. Some viral infections are meant to be spread only among animals, as you would find in stage 1, where the biologic agent is only such that animals can transmit it among themselves. You have bats, you have chimpanzees, you have monkeys and apes as common intermediate hosts for some of these viruses. By stage 2, we are talking about primary infections, where you have an infection being transmitted from an animal to a human, a susceptible human host. And when you talk about stage 3, we have these sporadic infections being spread among humans, but the index case must have had a contact with an animal source. And then in stage 4, you have more frequent outbreaks occurring among humans, yet the index case here still must have had some contact with an animal. In stage 5, the virus has evolved to the extent that it does not require an intermediate host, but it is now more stable in human population and continues to cause sustained human-to-human transmission. And this typifies the story of monkeypox, now redesignated Mpox. The natural history of Mpox is such that it's a virus that causes diseases in reservoir hosts, lower animals like squirrels and rats. And they could also transmit the virus to intermediate hosts like monkeys or African apes. And when monkeys are sold as exotic pet trade, they come in contact with prairie dogs, and those dogs are found in large cities. And so these dogs are mainly kept in families, and so they have contact with young children, young adults, and children. And if they are carrying the Mpox virus, they can transmit the virus, moving all the way from the rainforest across villages and towns to global cities where larger outbreaks would occur. But what typically occurs is that when an index case, maybe a cave explorer and hunter, or somebody that has some business in the rainforest, comes in contact with these animals, it becomes infected. When it goes back into the villages and towns, comes in contact with other humans, the disease is spread through close contact. And if you have one of such persons travel to a city outside of where they reside, and where you have more humans getting involved in closer, more intimate contacts, then you could have large outbreaks occurring in global cities where Mpox is not known to be endemic. So the story of Mpox is one that can be described as from neglect to resurgence. In 1970s to 1980s, there was a push to eradicate smallpox, and this was accompanied by a lot of mass immunization. And around that time, the first human case of Mpox was reported. By 1980, thankfully, smallpox had been eradicated thanks to mass immunization, and so the immunization stopped. Between 1990 to 2000s, there were sporadic outbreaks of Mpox, mainly in Democratic Republic of Congo and Central Africa. And between 2000 to 2010, it looked like the outbreak had started to grow. It was becoming more frequent, and a lot more number of persons were now involved. In 2018, we had our first major Mpox outbreak in West Africa, and Nigeria happened to be home to that outbreak. Some of the reasons adduced for the resurgence of Mpox included limited surveillance, limited testing capacity, and the fact that the antibodies against smallpox against the smallpox from the immunization that was done in large numbers in 1980, those antibodies had started to wane, and the antibodies were meant to be cross-protective for Mpox. And so with waning antibodies, reduced herd immunity, we started to have increased number of cases, increased number of outbreaks of Mpox. The Mpox outbreak of 2022 to 2023 was predominantly caused by the clade 2 lineage, or clade 2 of Mpox. We have two main clades, the clade 1 and the clade 2. But more recently, we have had more outbreaks caused by the clade 2 more than the clade 1. Clade 1 occurs more frequently in DRC as well as in Central Africa. And in the 2022-2023 outbreak, we had about 102,000 cases of Mpox reported globally across 103 countries, majority being from the United States, with about 220 deaths. 95% of these cases were in males that had sex with other males. And it seemed like the major mode of transmission was through sexual contact. And it also seemed at that point that the Mpox was displaying a kind of stage 5 level of infection that is associated with more sustained human-to-human transmission without any necessary animal contact, or history of travel to an endemic region. By July 23, 2022, the WHO Director General had declared the ongoing mpox as an outbreak of a public health emergency of international concern, and thus began a lot of interest in understanding why the mpox seemed to have evolved from a zoonotic infection to an infection that is now more stable among humans. At this point, I would like to warn that some of the contents in my subsequent slides might be a bit disturbing, and so viewer discretion is advised. I report a case, a 41-year-old male that had presented at our sexually transmitted infection clinic here in Ibadan with painful anal ulcer that was associated with purulent foul-smelling discharge. He also had some pustular rashes on the trunk, the soles of his foot, and the palm. He had an initial history of fever, body pain, and headache. There was no history of travel. He affirmed to be a male that had sex with other males, and he had had at least 3 partners within the last 1 year. He was screened for HIV and found to be reactive. He was also screened for varicella zoster virus and found to be co-infected. He was managed on outpatient basis, commenced on highly active antiretroviral therapy, and his symptoms resolved within 2 weeks. This is showing the some images from the patient, the perianal pustular lesions, and the pustular lesion that he had on his palm. He also had that similar pustular lesion on the sole of his foot. The next case also happened to be about the same age, seen at our sexually transmitted infection clinic, had presented with more extensive vesicular lesions around the genital region, as well as purulent urethral discharge. He was a newly diagnosed HIV patient, recently commenced on highly active antiretroviral therapy. He was a male that was also having sex with other males. He seemed to have multiple sexual partners, and he said he could not count the numbers because it was quite a large number. He also had some skin lesions on his trunk and his hands. We made an assessment of immune reconstitution inflammatory syndrome with query mpox. He was also tested for mpox, found to be positive. His varicella zoster test came out negative. He was admitted because of his extensive lesions. Unfortunately, the outcome in this instance was fatal. And these images showed the lesions that this patient had. You can see the extensive lesions on the thigh, as well as on the phallus, and also the urethral discharge. He also had some lesions on the back of his hand, as well as on his trunk. And if you are familiar with the stages of evolution of mpox exanthems, you would see that this is really at like the last final stage where you would be having some umbilication and some crusting. And so this appeared to be a kind of late presentation. Much earlier in 2018, with the mpox outbreak in Nigeria, my colleagues and I had called attention to the need for public enlightenment on mpox and the need for the appropriate infection prevention and control measures to be able to contain the outbreak. But as we started to have more cases reported across different clinics in Ibadan, from private hospitals and even from our dermatology clinic here at the University College Hospital, I realized it was time to study the mpox in more details. Thanks to the Global Virus Network Rising Star Fellowship, which had allowed me to be linked with my mentor at the Center for Virus Research, University of Glasgow, Professor Emma Thompson. And I also must express my gratitude to Professor Mayowa Owolabi of the University of Ibadan and Professor Pascal Mafa, who had facilitated my Erasmus Mundus scholarship travel grant that allowed me to be able to travel to the Center for Virus Research at the University of Glasgow in 2023 for about 8 weeks. And at that time, I had also sent some of the samples that we detected in our lab here in Nigeria. And that's a picture of me using the agreed ion nanopore sequencer to be able to do whole genome sequencing of the mpox. And the result from that study showed that all the clades that we had from our lab at that time were clades 2B, and they exhibited about 3 different lineages. Majority belonged to the A23, some belonged to the A3, and some were B1 lineages. For the A23 and the A3, they seem to show some novel nonsense mutations, which were compatible with the action of host ApoB3 enzyme. The implication of that is that those viruses have evolved to a stage 5 level where they are able to have a more sustained human-to-human transmission. And so it is not surprising to find that they were mainly transmitted through contacts, especially among some of our patients that were males that had sex with other males, and even those that were not MSMs, but had also gotten infected through contact with other index cases. For the H23, we found that the lineage seemed to have arisen about 3 years earlier than the time that we were detecting it, thus attesting to the fact that there must be some low levels circulation of these mpox viruses within our community, which is not being detected because our surveillance system is not robust enough. The B1 lineage has originally been shown to be a local lineage found that originated from Nigeria, but the one that we found designated here on the phylogenetic tree as KS3 seemed to cluster with the variant that had originally been isolated from France in 2022. And so it suggested that this was like a kind of back importation from the global monkeypox outbreak, which had occurred much earlier before we were able to detect this case. The data from our study, our collaboration with the Center for Virus Research, University of Glasgow, and also collaboration with the University of Surrey, has now been submitted in a paper titled Lineage Diversity, Genomic Mutations, and Clinical Features of Mpox Cases of Nigeria, 2022 to 2023, submitted to the Emerging Infectious Disease Journal, and we are keeping our fingers crossed for a positive outcome. And so that takes us to preparedness for the next pandemic. And I would say at this point that preparedness has to focus on being able to translate virology into public health action. And the only way we can do that is to bridge the gap between laboratory science, clinical care, by adapting epidemic intelligence, particularly in low-resource settings like Nigeria. The chain that starts out from detection to diagnosis, clinical management, integration of data, adapting epidemic intelligence, driving public health action into community impact, that chain must remain unbroken if we are going to be ready for the next pandemic. As a member of the Technical Advising Group on Virus Evolution for the World Health Organization, in the last 2 years that I've been a member, we've been able to publish 5 technical reports. 2 of them are basically on surveillance for the SARS-CoV-2 variants. Even though we seem to be out of the pandemic period, but there's a need to keep an eye on these viruses because they never will tell you when they are going to emerge and be able to cause large-scale outbreaks. And so currently we are monitoring the BA3.2 and the NB1.81 variants as they evolve. We are also looking at the Middle East Respiratory Syndrome coronavirus clades and lineages, and we've identified the existing gaps in knowledge, and we are proposing recommendations on how to be able to curtail the spread of this virus, particularly in the Middle East, where the spread of MERS-CoV seems to be associated with contact of tourists with the dromedary camels that are usually in abundance in those areas. Additionally, we are looking at, we've done a risk evaluation of the clade 1A monkeypox virus, reviewing evidence from all over the world and seeing how we can use this to contribute to policy statements on how to guide containment of the virus and how to prevent it from causing large-scale outbreak. More recently, we started to look at the Oropouche virus. Oropouche virus is a virus that is associated with meningitis and encephalitis, a kind of viral hemorrhagic fever, and which seems to be on the increase of recent in Brazil. And so we are looking at control measures, trying to look at the gaps in understanding and recommending the studies that needs to be done to be able to assess the public health risk that is posed by the circulation of this virus. More recently, the Technical Advisory Group on Virus Evolution of the WHO, we had come together to publish a paper in Nature Medicine that was describing a systematic nomenclature for the Mpox viruses that were displaying sustained human-to-human transmission. And as the member from Nigeria on that committee, had also contributed data from our lab in Nigeria. And we are currently using those data to improve tracking, communication, and public health response to human transmission of Mpox. There's a need to be able to integrate laboratory and field epidemiology. And the Nigerian Center for Disease Control has taken a positive step in this direction with the Field Epidemiology and Laboratory Training Program, which is done in collaboration with the African Epidemiology Network, HAFNet, and the African Center for Disease Control. And the whole idea is to provide a seamless linkage between laboratory science, which covers diagnostics, genomics, and pathogen characterization, as well as field epidemiology, which covers case detection, surveillance, outbreak investigation, and contact tracing. And when we do this, we'll be able to transform samples into data and data into evidence with actionable impact that feeds into global health. The advantage of integrating laboratory and field epidemiology is that we now have earlier detection of outbreaks before they can snowball into pandemics. We are now able to track variants as they evolve. We'll be able to trace the source of infection. Is it an animal source? Is this a source that we can find within the environment? This becomes easier with surveillance using genomics, adapting laboratory and field findings. And the data from laboratory and field findings would also feed into antiviral treatment guidance, as well as also evidence-based strategies for deriving the necessary vaccines that are required for prevention of different types of emerging and re-emerging infectious diseases. And this takes us to the 6 main points in pandemic preparedness. It all starts with having a robust laboratory and epidemiological system that allows for surveillance and early detection of viral outbreaks as they occur. There's a need to strengthen the laboratory capacity, ramp up capacity for sequencing, and also preposition personal protective equipment that could protect laboratory workers, and also have infection prevention control measures in place in laboratories, as well as standard operating procedures for different types of techniques that would need to be done. There's also need for health system strengthening, and we'll be talking about the need to have ICUs ready, the need to have oxygen pipes ready, and the need for training of healthcare workers on how they should respond to outbreaks of infectious diseases. We need to have a trusted means of communication that should be in existence even before the outbreak will occur. This is the only way we can counter misinformation, disinformation, and info epidemics as we usually have around outbreaks and pandemics. There's a need for emergency coordination and governance. We need to have an incident management system at the national level that can be escalated to the state, local government level, and which should be having regular simulation exercises and tabletop drills in preparation for when the real cases would appear. There's a need for research, innovation, and financing, partnership with industries who would help to translate knowledge on diagnostics, therapeutics, and vaccines into products that we can use very quickly in case we find ourselves in the face of an outbreak or a pandemic. It's important to talk about the One Health framework because we do know that most viral infections that would cause outbreaks always seem to have an animal host. And wastewater surveillance studies that have been done recently in most parts of the country, including here in Ibadan, Oyo State, where Professor Adeola of Microbiology Department is also championing wastewater surveillance for viruses and for bacteria of public health importance in collaboration with the Nigerian Center for Disease Control. We've been able to find that there's need for strong collaboration, communication, coordination, and capacity building in order to be able to monitor the 3 main sectors, the human sector, the animal, as well as the environment sector, and to be able to do that not just at the local level, but at the national level, the regional level, and the global level. And so the One Health framework is one that we need to adapt and the one that we need to implement. In resource-limited settings like Nigeria, we would definitely be faced with some challenges with pandemic preparedness because of the gaps in infrastructure and funding, limited trained workforce, shortage of supply chain for reagents and consumables for our laboratories, poor data systems, poor data storage systems, and lack of standardization of interoperability, as well as limited sequencing capacity, even in our existing molecular laboratories. And then human resource constraints has remained a major problem, no thanks to the JAPA syndrome. Some of the solutions and innovations that we propose include adapting portable sequencing, like the one that I described earlier, the suitcase lab by Professor Adeola et al., and having mobile labs that can be easily drafted for use on the field. We need to have a digital surveillance platform for tracking and monitoring our viral variants as they evolve. We cannot overemphasize the need to strengthen laboratory system, the need for collaborative research networks, both at the local level, the national level, and at the regional level. The African Center for Disease Control is doing a lot, but there's still need for improvement. There's need for public, private, and global partnerships in terms of development of new diagnostics, new vaccines, and then new therapeutics. And we also need to be able to do more research that will drive vaccination strategies across the globe. In closing, the next pandemic will come much sooner than we think. It might not take 100 years like we have had between the Spanish flu of 1918 and the COVID-19 pandemic of 2019. We are now more technologically advanced than we have ever been in the history of humanity. And so we should be more prepared. The world is more closely connected, but we're also more divided than ever, at least politically and ideologically. We are facing new challenges, which require new and better adapted tools. We are plagued with infodemics, misinformation, disinformation, and accelerating use of AI with its frequent hallucinations. Mpox remains a global public health threat that requires urgent action so it does not snowball into the next pandemic. Pox viruses seem to have become successful pathogens for human-to-human transmission, and the history of smallpox should serve as a lesson. History repeats itself when we refuse to learn from it. COVID-19 taught us a lot of invaluable lessons, but I wonder how many of that we can still remember at this time. Centering global collaboration, equity, and vulnerable communities should be the bedrock of future pandemic preparedness. At this point, I would like to acknowledge my collaborators here at the University of Ibadan, Dr. Olukemi Adekambi and Professor Moses Adewumi, my mentor and collaborator at the Center for Virus Research, University of Glasgow, Professor Emma Thompson, my former co-supervisor for my doctoral degree, now my collaborator, Professor Carlos Malukwa Demotis at the University of Surrey in England. I want to thank Dr. Dapo Adediji, our bioinformatician from Polesi, my mentor and collaborator, Professor Christian Happy, my colleague, my collaborator from Kwame Nkrumah University of Science and Technology, Professor Mohamed Mutuchele. And by no means the least, I wish to thank the staff and students of my department, the Department of Medical Microbiology and Parasitology, College of Medicine, University of Ibadan, and also the staff and students of Biorepository Clinical Virology, College of Medicine, University of Ibadan. And I wish to thank the Babcock University community. I saw a lot of them on this call, and I thank you all for keeping faith with me on my research journey. I conclude by saying, without local data, policies are driven by external assumptions. When we generate, own, and interpret our genomic intelligence, we shift from being recipients of global decisions to architects of our own health security. Thank you very much for listening to me, and I'll take your comments and your questions now. Thank you.
Thank you very much, Professor Fowotade. You can see the clappings, all the demonstrations of how people have valued your lecture. Thank you very much. Is Professor Rufus Akinyemi there, please? Professor Akinyemi? Please unmute, we can't hear you. Professor Akinyemi?
Yes, I was waiting to be unmuted.
Okay. Please, over to you. Thank you.
All right. Okay, thank you very much, Dr. Professor Fowotade. Our very own Professor Fowotade, but now Provost of the Carson College of Health Sciences at the Babcock University in Edition Remo. Thank you very much. We certainly didn't expect anything less. Excellent delivery. You sustained the entire audience throughout the entire course of the presentation. And of course, very stimulating and very cutting edge and exciting. Thank you so much for such an excellent talk. And of course, for putting Nigeria and Ibadan on the global map of virus hunters, as usually there too. Thank you so much. So we'll now take, and we want to thank our very, very esteemed audience. We have, the talk has drawn an audience of over 150, now about 150 participants, and I would say one of the largest audience in the recent history of these IAMRAT webinars. So we thank all our participating audience. That includes our elders, our leaders, drawn from far and near Nigeria and beyond the shores of Nigeria. Thank you all so much for being part of today's IAMRAT webinar. Permit me to single out our highly esteemed former Provost of the College of Medicine, former Vice Chancellor of the University of Ibadan, former Minister of Health of the Federal Republic of Nigeria, and an highly esteemed Ambassador of the Federal Republic of Nigeria, Professor IFA Adeola. Thank you so much, sir, for being out of your very busy schedule, being part of this webinar this afternoon. Thank you so much. Thank you, sir. So very quickly, we'd like to take comments and questions from our esteemed audience. Please, you may want to post your questions in the chat box while we take comments from, but if you want to speak, maybe make a comment or ask a question, please indicate by raising your electronic hand or post your comment in the chat box. Right, so I see the hand of Dr. Shakirat Godolufadi, Professor John Anator. So let's take comments from our very esteemed dermatologist, Shakirat. So please unmute and speak. And then Professor John Anator, Dr. John Olarewaju, in that sequence.
Thank you very much, Professor Akiyemi. It's
Right.
such an honor to hear you speak again after such a long time. And it's an honor, to attend this session by Professor Fowotade. Thank you for that presentation. There was something I was also going to mention, especially when you started to talk about the conditions, or especially those viral conditions that will present with dermatological manifestations. I recall that at a time, monkeypox was not thought to be a sexually transmitted infection until much later, especially when we started to see it amongst men who have sex with men. And so I wanted to mention that what I see with dermatological cases is that because dermatology is such a sanctuary area for a lot of people, and sometimes the things that should be scary to people are the things that are not scary, and the things that should not be scary are the things that are scary. So I would like to say maybe, I know there are a lot of lectures that have been incorporated dermatological wise. I'll mention a few cases like of monkeypox that I also saw. They would have presented, and initially most times they'll be misdiagnosed as either varicella or something else. And by the time they are coming, they'll be at the stage where absolutely nothing can be done. Or maybe they are at the stage where they have a lot of systemic manifestations. So I wanted to also mention that, just as Professor Fowotade reiterated, that dermatology is one of the ways that a lot of these viral conditions present. And maybe more lectures on things that will be scary or that should be brought to fore when it comes to presentations for this condition. And then I wanted to ask a question, and I think you mentioned it along the line of your presentation. Do you think, especially with the emerging and re-emerging infections, all these things that you talked about, do you think, I know it's not one cause, but if you take one cause of it, do you think it's the fact that we as humans are doing a lot of things that we should not be doing, or that we are maybe destabilizing the ecosystem of these viruses, maybe just kind of inviting them, like opening the door to these viruses? Is that, probably one of the main reasons why we're having a lot of these re-emerging infections? Thank you so much.
All right. Would you like to take a jab at that question?
Okay,
or
thank you very much,
all
sir.
the comments and then you can address that, right?
Okay,
Please go ahead.
Thank Dr. Shakirat Godolufadi, and to say that you are absolutely right. Most viral infections would present as viral exanthems, and so many times it's usually the dermatologist that is at the interface. And that's why it's important that dermatologists need to have a high index of suspicions. Some of the cases that I presented here, rather some of the genomes of Mpox that we sequenced were actually referred from our dermatology clinics. And that showed that the dermatologists were able to think beyond the box and suspect that this might be something that is likely to be beyond varicella zoster virus and most likely Mpox. That's the reason they sent the patient to the lab to get tested, and then we found out it was really Mpox. So we cannot overemphasize the need for having a high index of suspicion, particularly when we are within a very outbreak period. We must also increase our own alert to detection of these cases. As for the factors that are responsible for emerging and re-emerging viral infections, you are right when you mentioned some behavioral factors, like some weird behaviors, like males having sex with other males, moving into previously uninhabited areas because of increasing population, the closer interface that we are now having between animals and humans, leading to zoonotic infections and then spillover of zoonotic infections from animals to humans. And then climate change itself has not helped. And then the fact that viruses also seem to be able to evolve very quickly, mutate, and adapt from being a stage 1 pathogen to a stage 5 pathogen, which is more stable in humans. All these has helped as a kind of multifactorial cause for the reason why we seem to be having resurgence or re-emergence or even emergence of new viruses that were not in existence before now. So it's a combination of multiple factors all working together in favor of the viruses and against we humans as the hosts.
All right. Thank you so much for your excellent response. So may we now take comments from Professor John Aneto, sir, and Dr. John Olanrewaju. So we'll take their comments and then you also respond to the two comments, and then I will read out some questions from that box. All right. Yes, over to you, sir, Prof.
Okay, thank you very much. Good afternoon. Can you all hear me?
Absolutely, we can hear
yes,
you.
Prof, I can hear you, sir.
Yeah, congratulations for a very great lecture, Professor Fowotade, and congratulations also for your new appointment as the Provost of the College of Medicine at Babcock University. You will pardon my knowledge deficit in this area, but I just wanted to ask, is it possible to utilize the knowledge and skills so far acquired in this field to predict the outcome and scale down the risk of morbidity and mortality of health workers who are involved in handling situations like this when there's an outbreak? Thank you.
Thank you very much, sir.
Dr. Olanrewaju, let's take Dr. Olanrewaju's question or comment that you can address both together.
Okay.
Yes, please unmute Dr. Olanrewaju and ask a question.
Thank you very much, Professor Fowotade, for your topic this afternoon. This is the first time I'm having an opportunity to hear, to listen to talk on the monkeypox virus. So my question is this: from what you said, I discovered that the human-to-human spread of this disease seems to be more from male-to-male sexual intercourse. And I want to know, do we have such a situation in between a male and a female in a normal sexual intercourse as well? Then what are the other areas by which this disease spread? Maybe I didn't get that because I was a bit engaged while you were talking. So is there any other means by which this spread also from between humans? And my last comment will be, can there be more work and more effort towards precision medicine in detection and diagnosis towards this particular disease, especially from the question the first person who asked question was talking about, wrong diagnosis and people coming a bit late because it was not initially, identified that this was the particular disease they were having. So that's my question, ma'am. Thank you.
All right, you may please address those two before we take the next batch of two comments or questions.
Okay, thank you very much, Prof, for that question. And the answer to your question is yes. We can actually forecast outbreaks even way before they occur. And that is what predictive surveillance is all about. It's about combining our, looking at the data that we have in our gene banks, and then using the evidence that we have from the different surveillance systems, like wastewater surveillance that we do in the environment, surveillance that is being done, clinical surveillance among human beings, and even surveillance in animals. We can put all that together, do some mathematical modeling, and be able to predict that we are likely to have an outbreak of a particular disease at a particular time. And what that knowledge is supposed to be able to do for us is that it should be able to help us to be able to get more prepared in case these outbreaks could occur. And that is why we are talking about health system preparedness, strengthening the laboratory, building capacity, and then all the other things that I've spoken about during the course of the lecture. But the question is, in most low middle income countries like Nigeria, are we truly doing these things as we ought to? Are we actually prepared for the next outbreak? Everything that we seem to have learned from the previous outbreaks, from the previous pandemics that we have gone through, once we get past those periods, we put everything behind us. Hand hygiene is now relegated to a thing of the past. We are waiting for a time when there will be another resurgence, and then we start to wash our hands again. And then we've been talking about the issue of vaccination as a means of prevention, and a lot of people still are very hesitant about taking these vaccines, even when they are available, even when they are free. So all these factors make it very difficult for us to say that we are actually prepared for outbreaks. And the only way we can be able to reduce mortality, reduce number of cases from outbreaks, is if we're able to deploy all the available existing technology into preparing for these outbreaks. But I must say, unfortunately, that health system preparedness, particularly in Africa, in Nigeria, is still at very low minimal levels. And so most times we meet ourselves getting stranded in the middle of an outbreak or getting stranded in the middle of a pandemic. And even the things that we have had earlier on, those things degenerate and we do not build on them, and we start again from the scratch when we have another outbreak come through. With regards to Dr. Olariwaju's question about routes of transmission of the Mpox virus, before now, it was mainly known that Mpox predominantly is transmitted through contact, close contact, any form of close contact. And so it was only recently during the 2022-2023 outbreak that we were finding quite a lot more of this occurring among males that had sex with other males. It's not like it's something that is being transmitted by sexual intercourse, but it's actually being transmitted through the intimate contact that occurs during sexual intercourse. So it is still the contact and not necessarily the sexual experience itself that transmits the virus. But if you look at the way and the level of intimacy that occurs among males that have sex with other males, and the fact that they oftentimes share partners among themselves, that probably accounts for the reason why it looks like we're having a lot more of it among males that have sex with other males as opposed to heterosexual individuals. But that is not to say that it cannot be transmitted through close contact between a male and a female. It's actually the contact itself rather than the sexual intercourse that is responsible for the transmission. With regards to precision medicine is the way forward for almost any form of diagnostics, almost any kind of experience with regards to infectious and even non-infectious diseases. And so this is why there's a lot of emphasis on using the platforms that are available, the portable laboratories that helps us to be able to very quickly do real-time genomic surveillance that allows us to have better understanding of the kind of mutations, the points at which they occur in the sequences of the virus, and the implications that they have on the effects that the virus is going to have on humans. Like for our Mpox study, it was in the A23 variants and the A3 lineages that we found that they seem to have mutated at some point that has to do with the ability for them to be able to exist and be able to to undergo more stable transmission among humans that would not even require an intermediate host at all. So definitely precision medicine is very important in the outbreak prediction and is very important in the predictive surveillance that helps with preparing for the next pandemic. Thank you.
All right. Before I read out the comments and questions in the chat box, we'll take brief comments from our distinguished occupational health physician, mentor, Mrs. Professor Fowotade Omokodun. Please make your comments, ma'am.
Thank you very much. Provost, College of Health Sciences, Babcock, my dear Professor Fowotade, it's a pleasure listening to you. I actually joined this meeting on your 8th slide. I had a departmental meeting, but I was very interested to hear what you had to say. On Friday, this last Friday, we had NCDC give us a presentation on Mpox at my request. I actually wanted to know what was happening on Mpox. So when I heard you on Mpox, I was like, yeah, let me hear your version of it from a clinical virologist perspective. Because the NCDC is your public health arm in disseminating information to the communities and, that link between the lab and the actual community. And they actually told us that we had this spike in 2017, which you confirmed. And you alluded to the fact that smallpox immunity had waned, and that was why it was able to re-emerge. But they showed us a slide which showed that there was a peak in 2022, and then it went down again 2023, 2024, and another peak, but a lower one, in 2025. And I kept on wondering, what is responsible for these peaks? One of the possible causes is increased awareness, but is that the whole story? So I'm wondering from your perspective, what are the reasons that we have these peaks coming up? 2022 was quite remarkable, and then it went down again 2023. And the focus of the reports that they had was from the south of Nigeria. Do we have a lot of MSM in the south more than in the north? Is that why that is the focus? What are your thoughts about this? I just want you to give me your reflections as a clinical virologist. Thank you very much.
Thank you, ma'am. And your observations are very correct. I would say that with the 2022-2023 outbreak, there was a lot of awareness about Mpox, not just in Nigeria alone, but across the world, because this was a virus that was almost snowballing into the next pandemic, just when we were recovering from the COVID-19 pandemic. And so a lot of precautions were put in place. For viruses that are mainly spread through contact, unlike what we had with COVID-19, which was a respiratory virus, and which even when you put on your best practices, if you happen to be in an environment where somebody is coughing, sneezing, and you do not have your face mask properly fitted, you will still have the virus. But for Mpox, this is a virus that the main route of transmission is through contact. And so if you have a lot of awareness on hand hygiene, a lot of awareness on the need for limited contacts, particularly around the period where we're having an outbreak, that definitely will be followed by something that would look like a drop in the number of cases. But it always seems to me that our memory of preventive practices seems to be very short indeed. And even when we put on our best practices during the outbreak period, some moments or some minutes or some months or years down the line, we seem to forget about these things and go back to our usual practices. And when this happens, we would have a case that will feed on to another case, and then that case would, until we have like a chain that seems to be growing bigger and bigger each time. So I think the public health interventions that come in between those outbreaks are responsible for the reasons why we usually will have those dips. And then sometimes again, another thing that usually contributes to the outbreak is the fact that some cases get imported or back imported from places that are endemic. for those diseases, like the Democratic Republic of Congo, the Central Africa. Those regions seem to be more endemic for mpox. And so when you have travels across from those regions to other parts of Africa, other parts of the world, you can have those the viruses tracking back to other continents or to other countries and potentially causing infection among susceptible hosts who would then go on to spread the disease to their other contacts. It's not like we have more males that have sex with other males in the South than we have in the North or even here in the Southwest. I was quite taken aback too when I found that at least 4 of our mpox cases that we found here in Oyo State all affirmed to the fact that they were males that had sex with other males. And in collaboration with the Director of Public Health at the Oyo State Ministry of Health, we tried our best to be able to break into those cohorts, if only to be able to provide some kind of advisory that will help us to be able to break the chain of transmission. But it seemed like they had sworn to a oath of secrecy and would rather die than allow us to have some information on tracing their contacts or having some kind of interaction with their partners. They would rather stand and say, tell us what you need to tell us, and we will help you to disseminate the message. But they block every form of effort to try to link up with their contacts. So I think it's that kind of sexual practice is quite more widespread, even in almost every part in Nigeria than we really think we have going on. I think there's a lot of males having sex with other males all across the different regions of Nigeria. And it would seem that we just might not know, but we do have those clusters right around us. We've heard of cases of students doing that even in hostels. And so when you have these kind of things, definitely you cannot say that this is limited. You continue to have those things happening around us discreetly, except we have this outbreak of infections among them that will make us to know that we have this cluster and we need to be able to carry our public health intervention to wherever they are, because they would not come to us unless they become really very sick. So definitely we do have the MSMs. And I think the reason we are having those dips is because the public health interventions are working, but their memories are short-lived. And down the line again, we seem to have some reintroduction, some back importation. And this is the reason we continue to have like sporadic outbreaks all across different times and seasons.
Thank you.
All right, thank you very much. We'll take the last, I would say, oral comments from a very young, recent graduates and a promising researcher for the future, Dr. David Babalola. So over to you very quickly. After that, we'll then read out the comments in the chat box for our esteemed speaker to address. David, quick comments.
Yes, thank you very much, Professor Apiani. Thank you very much, Professor Fowotade. And I'd like to quickly acknowledge all my teachers here. It's been a fantastic presentation. Thank you so much. Your passion in how you teach your work has always been something that has inspired many of your students. Very quickly, Ma, in thinking about your comments, especially the more practical intervention aspects towards the end, you reflected on how we have to do so much in terms of strengthening our surveillance system and pandemic preparedness as a whole. And I'm just thinking that because of the way things work with epidemiology, you know, we are basically subject to the effects of damage from the weakest link in the chain. Do you think that there is any role that we can begin to look to the private sector to play? Well, I'm sure we all know the realities of things that happen with the state of governments and governance in our societies. And while we might eventually get around to having a public setup for some of these systems, whether it's laboratory and sequencing capacity or diagnostic innovations, I saw someone asked about RDTs or even, you know, bioinformatics, the infrastructure for genomic surveillance and things like that. Do you see any possibility that we can have a way to incentivize private players to contribute in the areas where we are weak now, so that we can, even if temporarily, you know, bridge those gaps? And then I imagine that there might also be ethical issues with this, because at the end of the day, there is that public good that we are trying to achieve by preventing pandemics. So how do we, bridge the gap with private sector involvement and then the possible ethical issues? Thank you very much, ma'am.
Thank you very much. And I do agree with you that we have a lot to benefit if we could have solid public-private partnerships that are long-standing and which are sustainable. I believe that this would help significantly with health system preparedness. I'm looking at the roles that public-private partnership will play potentially in the development of vaccines, in supporting development of new diagnostics, new therapeutics, which are all very important if we are going to be able to effectively deal with outbreaks as they occur. And I do know that some private partners, industries, for example, that do not necessarily belong to government, do have enough money to be able to help to support some of these processes. I'm looking into how we can have some health system strengthening being done from the private supporting oxygen supply in different institutions in preparation for when we would have outbreaks that would require patients that would need this oxygen. I'm looking at how we could have the private arm support research and some form of innovation into developing new diagnostics, or even helping to develop like apps that can be used for real-time tracking, for making developing real-time digital surveillance platforms that we can use for monitoring of emerging and reimagined infectious diseases as they evolve. The government is trying; they could still do more, but definitely with support from philanthropies, from the private sector, I believe that we will move further and faster than if we are reliant on the government alone to drive these systems. Thank you.
All right, so I will now summarize the questions that have been posted in the chat box from Dr. Ephraim Ogbainin, a mover from the Irua Specialist Hospital, is asking about the possibility of having rapid diagnostics for Mpox diagnosis. And then from our distinguished former director of IAMRAT, Professor Adinka Fadusi, she's asking whether the new knowledge that is being generated in this field is being included in current textbooks for training in African countries. And then from Professor Charles Iliku is asking whether students, if there are students who are being inspired to build career in genomics and global health, if there are specific skills and training pathways that we want to recommend for ECRs who want to build focus in that field. It's also asking if there is sustained funding apart from pilot projects and donor-driven initiatives. And then from Dr. Dotun Olalusi is asking if there's a role, if there's a place for CRISPR technology to apply to metagenomics and genomics surveillance in pandemic situations. And then from Dr. Abiodun Ezekiel additional is asking how can viral genomic surveillance systems be strengthened in LMICs? And in what ways can viral genomic data be effectively integrated into real-time public health decision-making without overwhelming policymakers? And if I may add the last question, that's what lessons have we learned from the outbreak of SARS-CoV-2 and ongoing outbreak of Mpox? The infrastructure that were built for SARS-CoV-2 pandemic, Ebola, and all the other pandemics, the infrastructure that were built in Nigeria and other African countries, are these infrastructures still there? The capacities, human capacity that were built, are they still there? Are we able to respond to the next pandemic with the lessons learned and infrastructure built from previous pandemics? So that'll be the last question. Will you please address them, and then I'll hand over back to the director. Thank you.
All the questions as you have read them out. Ones that you asked was whether there were rapid diagnostic kits for hemp.
Yes.
And I will say yes to that question. And just in last year, when I was working in Gambia, and I was volunteering as a consultant microbiologist at the MRC, Medical Research Council, the Medical Research Council Gambia unit of the London School of Hygiene and Tropical Medicine. I participated in a study that was ongoing where they were looking at piloting some mpox rapid kits that had just been discovered and comparing what the sensitivity and the specificity of the rapid diagnostic test kits to the use of real-time PCR for detection of mpox. And I also know that that was also being piloted in Senegal at the Louis Pasteur Institute in Senegal. So I believe that those data are pre-publication, and very soon we'll be able to see how the rapid diagnostic test kits for mpox has fared. Unfortunately, the rapid diagnostic test kits have not worked very well for Lassa fever, even though it showed good specificity, but the sensitivity seemed to be very low. And for a rapid diagnostic test kit, sensitivity seems to be a major quality assurance measure. So for you to say that you have a good screening kit for a particular type of disease. So we don't have the information yet on how the rapid diagnostic test kit has fared for mpox, but just to say that it's in existence and it's being piloted. To Prof. Fale's question about how this new knowledge is being incorporated into textbooks. I think just about 2-3 days ago, on the Committee of Medical Schools in Africa, we were talking about how we can have our own Africanized curriculum that would inculcate and include those local things that happen to be our own problems more than what we have in textbooks, which were made from, which we have been using, which mainly originated from the Western world, and which had more of the diseases that were not of interest or we do not often see in large enough numbers for us to be interested in. So I do agree with you, Ma, that we are getting a lot of new knowledge, even from our own local studies and our own local data here in Africa. And there's a need to start to think about how we can start to put our own textbooks together for teaching our students who are going to be practicing here back home in Africa and who are going to be faced with the kind of diseases that we are seeing rather than those ones that are seen in the Western world. For a student that is interested in developing skills and practicing as a clinical virologist or a public health virologist, I think for me, the doctoral degree has been very helpful because it was a good period for acquiring quite a number of hands-on skills that will be useful for making a diagnosis across quite a wide array of platforms available for detecting viruses. I think some knowledge of bioinformatics is also very helpful, and having some knowledge on epidemiology and how data can be translated into actionable evidence that can drive policy. I think that by itself is also going to be a useful skill to acquire for somebody who would be a virus hunter. And I know that there are quite a number of courses, some of them are even available free online that could be subscribed to, which will provide the necessary knowledge and information needed to be able to build up the necessary skills. For use of CRISPR in metagenomic surveillance, the answer is yes. And Dr. Olariwaju, who spoke earlier, who asked a question earlier on this call, is actually pioneering a CRISPR research group at the Babcock University, which is meant to see how that technology is a gene editing tool, to see how it can be deployed into development of new therapeutics, new diagnostics, and also its use potentially in vaccine discovery. So definitely CRISPR drafted into metagenomic sequencing would help significantly with novel virus discovery. With regards to the issue of lessons learned from COVID-19, mpox, and how this has fed into building capacity for diagnostics or even for clinical management here in Africa, specifically in Nigeria. During the COVID-19 pandemic, when there was a need to ramp up molecular detection for SARS-CoV-2, the then Director General of the Nigerian Center for Disease Control, Dr. Chikwe, ensured that we had molecular platforms for detecting SARS-CoV-2 spread out in at least 36 states of Nigeria. And some states happen to even have more than one of such facility. But what has happened between 2020 and the present time, 2026, is a story that I'm sure that we are all very familiar with. Most of those laboratories have actually become redundant because of the fact that maybe the coordinators of the labs were not innovative enough to think about how to utilize those laboratories for diagnosis of some other low-level infectious diseases, not just even viruses, but even for antimicrobial resistance surveillance and for many other uses that require molecular platforms for identification or for tracking of different genotypes of microorganisms. For those kind of laboratories, when the supply from the government stopped, it would seem that the life of those labs also stopped at that time. But it's good to also say that for some laboratories, there's still a lot of activity, a lot of effort, a lot of collaboration, some happening through research funding from non-governmental sources, from research grants. Some of those labs have continued to forge ahead, develop their molecular platform, and even expand the platform to be able to accommodate even some kind of biosafety level 3 pathogens that require a lot more biosecurity compared with the SARS-CoV-2 virus. So it's not a loss, a complete loss as it is. I would say that we have some labs that have continued to function, continue to grow, and have even now become recognized for detection of other pathogens. And so for those labs, one will give kudos to them. But for those ones that appear to be sleeping, it's important that they are giving some nudge so that they could also be more responsive and be more prepared, because we do not know when disease X, as the pathogen that will cause the next pandemic is now being called disease X. We don't know when disease X would come, and it will not be good that those labs would have run out of function by that time. So I do agree that this should serve as a wake-up call for all laboratories on the network. The Nigerian Center for Disease Control has a role to play. The African Center for Disease Control also would have a role to play in ensuring the sustained functionality of laboratories and the and continuous training and retraining to ensure that we continue to build capacity of the staff that work in such laboratories. Thank you.
So I just like to say a very big thank you before I hand over back to the director, Professor Kajai, that your talk has generated a lot of comments and questions, and that shows that you have stimulated the audience, and I'm sure that your talk has been very well taken. Thank you for reminding us that we need to be prepared for disease X, the next pandemic, and I'm sure everybody, all hands need to be on deck. Thank you, and I'll do you back to Professor Kajai. Thank you so much, Professor Fowotade.
Thank you very much, Professor Akinyemi, for the question and answer session. That was beautifully and efficiently conducted. I thank all those that have been with us since the beginning, and those who actually asked questions or shared their own experience. This has actually enriched the lecture. Thank you very much. I want to join Professor Akinyemi in thanking some of our senior colleagues too. Professor Falusi is the former director of IAMRAT, And I'm so happy she's on, and she's always been with us, supporting and also contributing. Thank you very much, Ma, for being at this lecture. We also have the Deputy Vice Chancellor of the University of Ibadan, who joined us, and that's Professor Arotiba, who was with us. And we have the former Vice Chancellor of Christland University, Professor Chinedu Babalola. Thank you so much for attending, and for our senior colleague professors that have retired, Professor Neto, Professor Bukunle, Bakari, and others. We're very grateful. To our collaborators and the networks of Professor Fowotade, I could identify some of them. Thank you so much for supporting her this afternoon. I'm sure she's very happy to really see you attending the lecture. And so also the people from Babcock. We are very happy to have you, and we hope that you will join us on other lectures, even if it's not from your provost or from anyone from you. So please, you are welcome, and we'll be happy to see you as we also give our other lectures. And I think I should also thank I think Professor Daibo was also on at some point, and Professor Daibo is now with WHO on leave. She's also a top-notch virologist at the College of Medicine, University of Ibadan. So I thank everyone for joining us. So let me come to Professor Fowotade. Professor Fowotade, I can't thank you enough. I mean, so suffice to say that I only gave you a few days, so to say, to give this lecture. And definitely, we didn't expect anything less. And you have really shown us what it is to be an expert and also a virologist for that matter, because viruses are something people run away from. But you faced it, and you faced it squarely, even in a pandemic situation. We thank God for your life. So just to say that someone posted something about Professor Fowotade, which I think I should share, because she actually demonstrated the brilliance that was accredited to her. This is one of her teachers when she was doing a residency in Ilorin. And he said she started residency training in microbiology and came to UCH for attachment. Bam! UCH grabbed her because she is very, 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 very brilliant. When she went to the UK, they also wanted to poach her. And well, for the love of our country, she must have come back. And we thank you for coming back. And you've done tremendously well. Thank you so much, and thank you. And we pray God will continue to enlarge your coast, and you will do more for this country, the continent, and the world globally. Thank you, and thank you. And for our team here at IAMRAT, we thank you. We thank you very much for being so cooperative, ensuring that these lectures are well conducted, right from Professor Akinyemi, Professor Adeola, and all other staff of IAMRAT. Thank you so much. So I want to promise you another exciting lecture next month, the last Wednesday of the month of March. Please block your calendar and be on the lookout. It's going to be an interesting lecture. Thank you, Professor Fowotade, again for the brilliant and captivating lecture. Stay blessed, and I wish everyone a good evening. Thank you so much, and bye-bye. For those who are still on, you may please register and also indicate if you need CME scores. This is very important. Putting it just in the chat is not enough. Please register before you go and indicate. Thank you very much. Professor Neto, thank you so much for joining. You've always been with us, and I can see that. So.